from Istanbul University, and the title of my speech is Chloroplast Genome Targeted Phylogenetic Analysis in Cucurbitaceae family. And let's first see the content of the speech about Cucurbitaceae metallurgical processes, two-dimensional and 3D analysis for phylogenetic assays with different softwares, and the conclusion section we can see. Cucurbitaceae is members of Cucurbitaceae are tuberous annual vines, and they're found in tropical and relatively warmer regions. In generally more than 800 species are present for Cucurbitaceae family, and the members have rarely toothed leaves and palmate veins, and each leaf has a combination of branch growing on one side, a vegetative bud, and female flower and an inflorescence. The branches of the Cucurbitaceae are part of branch complexes and may or may not stem some branches, and some botanists does suggest that these species are modified, including modified shoots, and many Cucurbitaceae have nectar discs, and pollination by bees is very common, and the topic of the speech is related about limited information about the intraspecific or interspecific variation for the members of the Cucurbitaceae family. And we see some common samples from our kitchen belonging to the Cucurbitaceae family, and we try to get knowledge about interspecific variation from the species selected 10 predominating species of Cucurbitaceae family by using nucleotide sequence data from gene bank and related databases by targeting chloroplast DNA analysis. In material methods section, here we see sequence data for 10 common species for Cucurbitaceae family belonging to chloroplast DNA, cpDNA. And the first metallurgical process is related to the GraphPad Prism software that we use for obtaining correlation matrix and principal component analysis. Here we see some details and the criteria for different species of Cucurbitaceae family, the origin, soil taste, and the sizes for and numbers of the specific genomic characterization, and the remaining species are also listed in this table. And we go to the results from the GraphPad Prism software, and here we see the firstly p-values and the dimension values for print correlation matrix, and we can only say that there's a positive correlation between percentages of cytosine guanine percentages and number of the mRNA, and the number of mRNA or cDNA increases may be related to the GC percentage rate increase, and here we see it from the B plot of Prism. And also we can say that specimens which are grown in loamy soils are clustered together, and also Cucumis mellow show this very distinct profiling from remaining species, especially by geographic origin, soil characteristics, and the taste. We can easily follow from the plot analysis. And the second analysis were carried out primarily mapped software. Sequence similarities and UPGamma-based trees were constructed using Genus Prime and MAFT software. FASTA files were converted to Align, Philip, and Nexus files by using online tools, and then subjected to the common terminal commands of the MAFT software. And then we get UPGamma-based dendrograms, and we can easily follow that species of the same genus were co-clustered in the same sub-branches from the two different dendrograms. We can easily follow them. And the third methodologic process is is coming from Linux terminal, Mr. Bias. We also use Align and FASTA files to convert Nexus file by Phylolab, and then Nexus 5 were subjected to the Bayesian topology by using Mr. Bayes software from Truba Linux, and then phylogenetic trees were obtained using Fig3 software with the common comment, and then we obtained collapsed profiling with different species we are belonging to the different side of this collapsed phylogram, and the similar results for the character-based dendrogram, such as in distance-based dendrogram from the previous results. And another assay for phylogenetic analysis by RStudio was carried out using three different packages to construct phylogenetic trees, and we obtained maximum likelihood and distance matrix analysis, and then we obtained the finally principal component graphic with great level of variation. The first axis PC1 is about 51.2% with the great level of variation, and the genetically most similar species were belonging to the same genus, while the genetically most distant species were belonging to the different genus, as we see Cucumis and Momordicata. As a conclusion, in the study, several different botanical approaches, biostatistical approaches, were used to examine the interspecific variation for cpDNA of Cucurbitaceae members, and the positive correlation was obtained for the mRNA number and the GC percentage among the CP genomes. And Cucumis melon appears to differ from other species in more than one characteristic. No distinguishing features for other species, but C. melon for cpDNA and other soil characteristics. In PC analysis, there is no clear distinction for remaining other species, but we can easily say that C. melon is going very different from other species in principal component analysis. As in common previous polymorphism studies, cpDNA could be assigned as able to discriminate between the genus strongly intraspecific variation. In this content, it is thought that marker genes included in the cpDNA may be identified and used in further studies for distinguishing and distinction of Cucurbitaceae family members. Obtained data suggests that the sequence diversity of cpDNA, a strong phylogenetic analysis material is on-site, even part of the characteristics of data of Cucurbitaceae. And here we see the thanks and the reference section as a last slide. Thanks much.